All right, so in prophase of meiosis 1, we have the chromosomes have been copied and condensed. The copies are the sister chromatids, like we saw before, attached by a centromere. And then the homologous chromosomes here are shown as different colored chromosomes that otherwise look pretty similar. And unlike mitosis, during meiosis in prophase, we will see these homologous pairs come together. So this is like the number 1 chromosome that this cell had inherited from the father. And this is like the number 1 chromosome that the cell had inherited from the mother. And now there are 2 copies of each one of those, the 2 sister chromatids on each of those chromosomes. When those 2 homologous chromosomes that are now really duplicates, the 2 sister chromatids on each one, when those 2 come together, it's called a tetrad. And this happens during meiosis because an important step happens during this joining of the homologous chromosomes. And this important thing that happens is called crossing over. When the homologous pairs match up, if you look here now, we have written some genes symbolized by these letters A, B, C, D, E. And if we look on the 2 homologous chromosomes, we see that on the 2 sister chromatids, those gene designations, the letters, look identical. Capitals all on the 1 chromosome, lowercase all on the other chromosome, but A and A, B and B. Same genes, but different forms of the genes, different alleles. Well, organisms who reproduce sexually have evolved this crazy step called crossing over that increases genetic variability. It increases the differences between offspring. This is a great idea in terms of evolution because when organisms reproduce sexually and produce offspring that are quite different from one another, that's like producing lots of different strategies for surviving in an ever-changing environment. So many species, most species, have evolved sexual ways of reproducing so that they produce offspring that have a variety of different genetic makeups, which means a variety of different genetic strategies and ways of surviving in the environment. And what happens during crossing over is the homologous pairs match up, and then groups of enzymes come in and cut out big chunks of chromosomes and swap them between the 2 homologous chromosomes. So now we see on this chromatid, on the chromosome on the left side here, that it has the A and the B, capital A and B allele, and then has lowercase c, d, and e. That combination of those genes did not exist on the chromosomes before that. Every time a cell goes through meiosis, these random swapping of genes happens, and you wind up with a chromosome that nature has never seen before. A brand new genetic book for guiding the production of a cell or of an organism. And so crossing over takes place during this phase of meiosis 1, and it's a way of creating brand new collections of genes on chromosomes by swapping out homologous genes on homologous chromosomes. The rest of meiosis 1 is going to look a lot like mitosis. Those homologous pairs line up across the center of the cell, sort of the roll call of the cell. And what is different in this phase is that during mitosis, along the equator, all the chromosomes would just line up and you wouldn't see these homologous pairs. As anaphase begins, the homologous pairs get pulled to opposite sides of the cell. And at the end of meiosis 1, in telophase and cytokinesis, we wind up with 2 daughter cells that now are haploid. One of the homologous pairs is in this cell, the other in the other. One of the second homologous pairs of chromosomes in this cell, the other in the other cell. So there are 2 chromatids for each one of those homologous chromosomes, but we've essentially gone from diploid to haploid in this first division. And then the cell completes division with cytokinesis, and it's ready to do the second division, meiosis II. Now we have the cell that has sister chromatids still attached at the centromere, but the pairs of homologous chromosomes have already separated from one another. In this part of meiosis, the second phase of meiosis, the cells are going to do a process that looks very much like mitosis again, and this time we're going to separate the sister chromatids. And here's what that looks like. Here's those cells just dividing that we saw in the previous diagram. Here we go with the spindle forming and the microtubules grabbing those chromosomes, lining up the chromosomes across the middle of the cell in metaphase, separating the sister chromatids during anaphase, and then completing the second division in telophase and cytokinesis. And now we started with a cell that had 2 pairs of chromosomes, 4 chromosomes in total, 2 pairs of homologous chromosomes. Now each of the 4 daughter cells has only 2 chromosomes, 1 each of the homologous pairs. Here are a couple of slides that show some details of those stages of meiosis II. Pause this and read these as you see fit and as you need. And then what happens to those cells when it is a male producing sperm? Well, all 4 of those daughter cells that are produced by meiosis become sperm cells. Each one differentiates into a little squiggler, and the nucleus in each one of those is going to be haploid, just like those cells that produced the sperm. And in females, this almost always looks different, and it almost always looks like this. Eggs need to be big because they need to have enough stored nourishment to get the embryo started in life on its own. And so when meiosis takes place in females, the cell divisions are unequal. The first division and the second division of meiosis, one of the cells keeps virtually all of the cytoplasm. And that means it will make a big egg cell and 3 smaller cells that normally play no role in reproduction. And those are called polar bodies. And really the function of the polar bodies is just to take out the extra chromosome so that you wind up with a haploid egg. So again, this unequal division of the cytoplasm is happening so that you have a single haploid egg that's large enough to have enough stored nourishment to give the embryo a start in life if that egg happens to be fertilized. Here's a nice comparison of both processes side by side. In this case, it involves a cell that has a diploid number of 6 chromosomes, so 3 pairs of homologous chromosomes, and I like the way this comparison lines them up side by side. So pause and take a look at this and see if this is making some sense for you. Again, I wrote it out here. I don't want to read it. Pause this and read over this and see if these similarities and differences between mitosis and meiosis are aligning with what we just talked about and what you're learning. And here are some questions for you to check your understanding. Feel free, and please do, go back at any place in the video to help you reconnect with some of these ideas and see if you can answer all, or at least as many of these questions as you can. And any of these questions that don't seem like something you can tackle, that's a question you should ask when you come to class next time, or when you Zoom in next time.